Good morning and good afternoon, everyone, and welcome to this latest Nano in Action webinar. My name is Chiara Venturini. I'm the Director General of the Nanotechnology Industries Association. And before we jump into the subject matter of today, which is the applications of nanotechnology in agriculture, I would like to give you a very brief introduction of the NIA and our activities. We are a membership association based in Brussels with offices across Europe. And our main mission is supporting the successful commercialization of nanomaterials. In order to do that, we support our members in a number of activities. First and foremost, the understanding of what kind of rules they have to comply with in order to place their products onto the market. The establishment of trust from customers and consumers on the safety and performance of their nanomaterials. And because the nano industry is a very varied environment with a number of very diverse applications. We also support with the identification of the right target audiences and potential end users for the different applications. Our activities can be grouped in three broad clusters with support with the establishment of the appropriate regulatory environment for nanomaterials to successfully be placed on the market. And we do this through a number of activities, including regular interactions with policymakers at national and international level. We provide a platform for our members to reach out to other actors in the nano community and also to showcase their products and services. And finally, we support the establishment of the right ecosystem for the scale-up and uptake of nano products. The complete list of our members is on our website. So this is only a snapshot. I just would like to stress that our membership covers the whole spectrum of activities in the nano environment. So we don't only represent manufacturers, but we also speak for the providers of collateral products and services. And we also have a strong R&D component with a number of research institutes and universities among our members. I wouldn't read bullet point by bullet points the list of activities that we implement. This is, again, just a snapshot. I would like to stress that part of what we do is exclusive to our members that receive regular briefings, updates, and have regular opportunities for engagement with the activities of the association, but we also publish a number of materials for the broader public. Just last week, we released a position paper on the evolution of the language to define nanomaterials and nanoforms. So I would invite all of you who are interested in learning more to download the paper from our website. The event that you are attending today is a good example of that platform that I was mentioning earlier on. One of our members will speak in a moment. And of course, as most industry associations over the last year, we have had to move most of our activities online. And we foresee that for the remainder of the year, this will probably still be the case. So keep an eye on the event section on our website to have an idea of what is going to come in the next few months. Of course, to remain up to speed, you can also register for a monthly newsletter. We are active on a number of social media. And if you would like to learn more about not only how you can remain up to speed with the information, but also to steer the activities and potentially to suggest new ones, I would be very happy to discuss with you how you can get more involved and discuss membership options if that is of interest. With that, I would suggest that we go straight into the topic that we'll discuss today. So I will just stop sharing my screen and introduce our first speaker for today, who is Dr. Chaturika Surani, who is a senior scientist at one of our members, SLINTEC, the Sri Lanka Institute for Nanotechnology. So Dr. Surani, the floor is yours. I see that you have already put your slides on the screen, so I would just hand over to you. Thank you.
Okay, thank you, Chiara, for the nice introduction, and hi, everyone. Today, I will talk about the nanotechnology application in food industry. First of all, I would like to introduce my institute, Sri Lanka Institute of Nanotechnology, which is located in Sri Lanka. It is a private company formed through the public-private partnership between the government of Sri Lanka and 8 leading private sector companies. Our mandate is to build a world-class research and development center specialized in nano and advanced technologies, and to make products more competitive using nano and advanced technologies to the society, and added value to the Sri Lanka's mineral resources by showcasing the benefits of coupling minerals with the nanotechnology, and to build a nanotechnology and science park for research, development, and commercialization of innovations. Our main focus areas are sustainable agriculture, smart textiles, medical diagnosis and preventive medicines, energy, minerals and composites, and also the printable electronic and sensors. What SLINTEC can offer? Actually, we have a very latest equipment, including XPS, SEM, TEM, and NMR. And also we have a highly trained scientists. With these two together, we can embracing the cutting edge technologies into the society. And also other companies has provision to incubation space to locate their R&D facilities, and also pilot scale production facilities at SLINTEC. And also those other companies have opportunity to create value by outsourcing R&D without costly capital investment as well. Okay, now I will move to my actual topic, nanotechnology in food industry. First of all, we will look at what is nanofood. The term nanofood describes food which has been cultivated, produced, processed, or packaged using nanotechnology techniques or tools, or to which manufactured nanomaterial have been added. I want to stress here, cultivated, produced, processed, or packaged. If your food is cultivated, produced, processed, or packaged using nanotechnology or their applications, that food is called nanofood. In the food industry, nanotechnology is applied for all practices like production, processing, distribution, and protection. I will discuss about the food processing, distribution, and protection. The third speaker, my colleague Ruchira, he will discuss some of the applications related to the food production. First of all, I will go to the application of nanotechnology in food processing. The post-harvest processing has immense potential in nanotechnology applications. So, in the post-harvest processing technology, the bioavailability of the food is the nanotechnologies in applications can be used to enhance the bioavailability of the foods, the taste, the texture, and the consistency of the foods. And also, this technology can be used to remove the unpleasant taste or odor in the food during the food processing when you process your food. And also, we can modify this particle size distribution using the nanotechnology for food processing application. The edible nanocoatings are also used in food processing with the nanotechnology applications. Uh, this can be applied. The edible nanocoatings are applied to perishable products such as meat, fish, fruits, and vegetables. This coating serves as a gas and moisture barrier, and also additionally can provide the flavor and color or enzyme to the product as well. At the same time, with the barrier properties, it can prolong the shelf life of food as well. As examples, beeswax coatings and pectin are widely used as edible nanocoatings in food processing industry. The healthier foods. We know that we have these days we have these foodborne diseases. To develop healthier foods, which is containing low fat, low sugar, and low salt, the nanotechnology is nanotechnology are being used. So, this can increase for the bioavailability of the vitamins uh, in the food supplements. Like we know that the iron and omega-3 fatty acids are low in bioavailable. So we can use nanotechnology, we can use the nano encapsulation, nano emulsion to increase the bioavailability of these iron and omega-3 fatty acids. Another application in the food processing industry is antibacterial coatings. Antibacterial coatings are again used to prolong the shelf life of perishable products and avoid contamination of the pathogens. The naturally derived nanoparticles, which has antibacterial properties, can be used as edible antibacterial coatings. And the nanofilters also used in the food processing industry to remove colors in some of the food processing industries. In the oil industry, nano clay is used to remove the pigments in the oil. Nano sieves used in the milk industries to remove the lactose in the milk and making milk suitable for lactose intolerant patients. And I'm talking like nano emulsions, nano encapsulation, like first. So first of all, we will look at what are these nano emulsions, nano encapsulation, which can be used in the food processing industry. Nano emulsions are colloidal particle systems with oil in water. Like in here, you can see in this picture, the left side of the picture. This is the emulsion of water in oil in water, and this is the water in oil emulsion. We can use these emulsions, nano emulsions, in various applications in food processing industry. These nano emulsions are very small in size, ranges from 10 to 1000 nanometers. These nano emulsions have excellent properties like high optical clarity, high stability, and high bioavailability. The advantages of these nano emulsions are with this small size of the emulsion, they have large surface area, which is very important for strong interaction with various bioactive compound transport into the target specific cells. It has higher digestion rate, where it has more binding sites for digestive enzymes in the gastrointestinal tract as well. Other than that, the nano emulsions are help in rapid transfer of naturally occurring hydrophobic bioactive compound in functional foods as well. The nano encapsulation. Encapsulation can be used as a nutrition supplement, and also it can be used to remove the unpleasant taste and improve the bioavailability of the some of the foods. And also it permits efficient dispersion of the insoluble supplements without surfactant or emulsifiers. The encapsulation can entrap biologically active components like vitamins, antioxidants, phytochemicals, proteins, lipids, and carbohydrate to improve the function of the food and the stability of the bioactive compound. And this picture shows you the few example of nano encapsulation, which is used in the enhancing of the bioavailability of the curcumin. We know that curcumin is low in bioavailability. So we can enhance the bioavailability of the curcumin by encapsulation techniques. There are few encapsulation available. These are few examples like polymeric micelles, polymer nanoparticles, nanogels, dendrimers. Those are the few examples for the encapsulation. Then I would like to move on to the food packaging technologies. The In the food packaging technology, active food packaging is one of the main category. These food packaging systems consist with the moisture regulating agents, carbon dioxide scavengers and emitters, oxygen scavengers and antimicrobials. Uh, and in the active food packaging system, nanocomposites are widely used. So in in here with the nanocomposites, they can enhance heat resistant, low weight, and mechanical strength. Likewise, the nanoparticles such as silicon dioxide, silicate nanopellets, carbon nanotubes, starch nanocrystals, graphene, chitin, cellulose-based nanofibers, those are used in the polymeric matrix to produce nanocomposites. You can see here, you can use layered material or nano particles with the polymer matrix to produce the nanocomposite, polymer nanocomposites. This, this incorporation with above nanoparticles or the nanomaterials in a polymer matrix, the matrix become lighter and enhance the heat resistance, mechanical strength, increase barrier against oxygen, carbon dioxide, moisture, UV, etc. And also another packaging system is called modified atmospheric packaging system. It is also important packaging system where it prevents a reaction of the reach of the oxygen, carbon dioxide and moisture to the food, and also with the same time it can enhance the shelf life of the food. The other packaging system is antimicrobial food packaging system. This antimicrobial food packaging system also widely used in the food industry. Perishable foods are more susceptible for the microbial damage, and these antimicrobial packaging are being used for the perishable food industry. The metal and metal oxide nanoparticles incorporate into a packaging material, and we can achieve the antimicrobial properties of that packaging material or that package. Iron, silver, uh, silicon dioxide, zinc oxide, carbon, magnesium oxide, titanium dioxide are widely used as antimicrobial agents in a food packaging system. As example, the titanium dioxide can produce reactive oxygen species which can detrimental for the pathogens. And other food packaging system is intelligent food packaging system. This packaging system designed for sensing the biochemical or microbial changes in the food. And also this system, they use nanosensors to detect the specific pathogen developing in the food or specific gases evolved in the food spoilage. Other interesting topic in the food packaging technology is edible food packaging systems. In this approach, they use some of the material like cellulose, starch, SAIN. These materials synthesize as nanofibers and used to produce edible food packaging systems. These packaging systems have superior properties like it has increased heat resistant, increased barrier properties and high thermal stability, and also we can incorporate a flavor to the material as well. That is an additional advantage. With all of that, I will move to some of the our work which we have done at SLINTEC, Sri Lanka Institute of Nanotechnology. We have produced few products related to the food technology using nanotechnology applications. First, I will go into the product that we have developed to increase the shelf life of fruits and vegetables by using ethylene scavenging material. In here, what we have done was we encapsulated ethylene scavenging compounds into a nano microparticles, which is naturally occurring. And with that, we promote sustained release of encapsulated ethylene scavengers for prolonged shelf life of fruits and vegetables. And we obtain 30% of delaying in ripening. In these products act in a 3 different ways. It can absorb ethylene, it can oxidate ethylene, and it can absorb the moisture as well. And another 2 products, that is nano composites, which produce with the low density polyethylene. With the first one is with the MMT films, and the second one with the graphene oxide films. In these packaging materials, they have obtained water vapor transfer rate improved by 20 to 50%, and they improve the stiffness, and exhibits the excellent dispersion properties and high UV barrier. And these 2 packaging systems can be applied for all packaging application to increase the shelf life, and especially the graphene based one, it can be applied to anti-static packaging materials as well. Thank you very much.
Thanks a lot, Chaturika. I have seen a question popping up in the chat. Just as a reminder for those who may not have joined any of our webinars before, we will take the presentations one after the other, and we will keep the questions for the Q&A at the end. If you're like me and have very short-term memory, note down your questions for the Q&A. So I'd like to hand over to our next speaker, Dr. Lucy Vyslogilova, who is International Project Manager at Nanoprogress. Lucy, whenever you are ready to share your screen, feel free to go ahead.
Thank you, Chiara. I try to share the screen. I hope you can see my presentation.
We can see it. It's not in full screen mode yet, but we can see it.
Perfect. Thank you. Okay. Good morning, everyone. My name is Lucy Vyslogilova. I am a cluster manager at the nanotechnological cluster organization Nanoprogress. And today I would like to introduce you our development, our research about nanofibers, nanotechnology, and especially I would like to show you some nanotechnological application for agriculture and healthier environment. At the beginning, I would like to say some words about our cluster organization, Nanoprogress. We were founded in 2010, and we developed our first equipment in 2012, and it was the first equipment in the world what was able to prepare industrial production of core shell nanofibers. They are nanofibers, bicomponent nanofibers with some shell material around the core one, and we are able to prepare material, for example, in some liquids, with some drugs, with some growth factors, and so on, as the core part of the core shell nanofibers. In 2014, we won the best enterprise project in the collaboration of companies and clusters category. We continue with research and development of nanofibrous-based structure, and with equipment to produce these structures. And in 2015, we developed the first AC electrospinning equipment. What is it? AC electrospinning is unique technology for production of nanofibers for different applications using alternating current. So we produced our first equipment and we continue with our research and development. In 2016, we won the Gold Cluster Awards for a most dynamic cluster organization. In 2017, we won the Gold label. And in 2018, Nanoprogress, with 6 other European partners, established ADPAC. What is it? ADPAC is EEIG legal structure, and we won European Strategic Cluster Partnership of the Year in 2019. In 2020, we defended the Gold label, and in 2021, we developed the first AC line for electrospinning of massive production, or it was equipment for massive production of nanofibers. We developed this equipment with Nanotech Dynamics company, who is one of our members. And this electrospinning equipment is able to produce massive nanofibers in 1D, 2D, or 3D structure for different applications. And now I am not sure, but I can move with my presentation. Now, sorry. Here you can see our research facilities. We have, at the moment, we have 3 research facilities in Czech Republic. 2 of them are in Buštěhrad and in Roudnice nad Labem, and they are facilities for basic research. Our basic research and also applied research is focused in Liberec city, and here we have our material laboratory for material characterization. Here you can see our laboratory and pilot equipment, and we center our research especially here. Because we have big plans and vision, you can see in 4 pictures, you can see our future vision, our future concept. This year we bought this very old building in Jeřmanice city, and we would like to build new research center of Nanoprogress, and we would like to focus our basic and applied research with equipment for classic industrial production and also for medical, for clean production of nanofibers here in this building. By here, it's a small city, but it's in the strategic location. This city is on the road from Germany to Prague, and it's around 20 minutes from Germany. Here you can see our main focus. We are focused especially for 4 main domains. We are focused on R&D support and technology development, on product development. In the picture, you can see our colleague, and he is in our laboratory in Liberec, and he is there with our 2 equipment developed by us for electrospinning of nanofibers. We are focused also on added value services for our members. We offer our members share infrastructure. We offer support with project preparation, administration, and realization. And our 4th domain is internationalization. We would like to connect our members with other company and institutes from European Union and also from all world. And now here you can see our developed materials, our product, who are able to apply for different industries. The first of them is a nanofibrous-based candle filter. We are able to prepare products based on nanomaterials for advanced filtration. What it means. Here you can see some white candle, and it is combination of classic yarn, and this yarn is covered by very fine nanofibers. So it means we are able to filtrate very small particles, bacteria, even virus, and we are able to use this candle filter in different applications. Here you can see what we are able to do with this filter. We are able to apply this candle filter in industry application, food application, and in agriculture application. And when we started our research with these filters, we cooperate with our partner in wine industry. And we tried to filter red wine with our nanofibrous-based filter. And what happened? We filtered red wine and we obtained white wine. But what was more important is the color changed, but the taste changed only a little bit. So it was very surprised for us. So it's our funny story about filtration. We are able to use this special filter not only for filtration of wine or beer, but also in cosmetic, for example, in filtration of different type of oils. We are able to use this, and we develop these filters also for pool filtration, and we are able to filtrate not only wastewater, but also we are able to filtrate drink water or air filtration and so on. Our next product, what we developed with Technical University of Liberec and with company Nanotech Dynamics, is biomass carrier. What is it? What you can see in the picture are nanofibers with biomass. What it means? We are able to prepare special systems for clean water. We are able to treat wastewater and also capture the phosphor or drugs or other pollutants from the wastewater. It's very important because it's not so easy to capture the pollutants and drugs and phosphor from the water, and there are limits of the water in the planet, and we would like to prepare some system, or we prepared some system what is able to prepare clean water. Here is our next application, and it is nanofibrous sorbent. It's a material for oil sorption. This material or sorption of this material is 200 times higher than classic material for sorption. And also we are able to regenerate it this filter is able to repeat it regeneration. So it's very important, and it's suitable for oil or fat sorption. We also focus not only more industry and agriculture's materials, but also on food materials. Here is our product based on nanofibers, and we are able to detect shelf life of food. Here are different colors of nanofibrous sensor, and we are able to see if material, for example, salmon, is fresh or not so fresh, or absolutely not so fresh. And it's important for waste of the food, because today more than 60% of produced food is waste. And we would like to be more familiar with our planet, and we prepared this special nanofibrous based sensor who is able to say us if the food is fresh or not, and if we can eat it or not. And we developed this material for fishes, but we are able to prepare material also for seafood, cheeses, dairy products, reduction, and it's for reduction of food waste. So it has a lot of different way to use it. And that's it. Thank you for your attention.
Thank you very much, Lucy. And as I mentioned, keep any questions you may have for the Q&A. While we go on to the next speaker, and we go back to SLINTEC with Mr. Ruchira Vijayasena, who is also a senior scientist at the Sri Lanka Institute for Nanotechnology. And Lucy, you can stop sharing your screen now. And Ruchira, whenever you are ready to share your slides, please go ahead. And you may be on mute.
Hi, can you hear me?
We can hear you now, and we can see your slides, although not in full screen mode yet. Now we can. Yes, thank you.
Okay. All right. Okay. Good morning, evening, ladies and gentlemen. My name is Ruchira Vijayasena. Today I'll be giving you this presentation on nanotechnology applications, particularly in fertilizers and in soil nutrient management. So agenda for the presentation is very simple. We go to a small introduction to nanotechnology, and then we talk about nanotechnology for fertilizers and how we can increase this fertilizer use efficiency, and also how we will then go into soil nutrient management, especially how we check the soil nutrient state of soils using various advanced technology related methods. Okay, so let me start with the nanotechnology. There are three main reasons why we have this interest in nanotechnology. Primarily, especially the nanoscale is a very interesting size scale of matter, because at this scale we have the emergence of both bulk properties and nano properties. Especially as we know, in bulk materials, they are particularly governed by Newtonian physics, and the atomic scale materials, they are particularly governed by quantum physics. This interesting scale between 1 nanometer to 100 nanometers, where this whole physics tend to blur. So the main, the reason that when you work with at this particular scale, you can actually tune properties to get tune matter to get specific properties, and also we get a high a specific surface area, and also this is the scale that biology works. So these are the three main reasons why we have we use nanotechnology. It has a high ability to work with basically tuned materials for our applications. Now, if I specifically go into this, the first reason, the main reason. Now, for an example, if I take a silver, we all know silver is a shiny whitish metal, but it's a very characteristic property of this metal. But when you go down to nanoscale, what you can, especially if you make a water dispersion of silver nanoparticle, they tend to be different in color. They appear yellow in color. Gold is similar. If you get a gold nano dispersion, suddenly the gold, the color, the characteristic yellow shiny nature goes into reddish color. So what you can see here is that there's a characteristic property change. So not only the color is a very basic property, but there are also other properties like the reactivity, the melting point, electrical conductivity, all these properties can change. So what does it mean? This means that we can tune materials to get specific properties for our applications. And for agriculture, and primarily there are two specific ways that people use. People use nanotechnology in terms of nanocomposites. So basically here we add a little bit of nanomaterials with specific properties to bulk materials to get bulk materials with interesting properties, properties of nanomaterials. That's one way of using that. The second method is basically nature does this best. We have nanostructures, especially lotus leaves, the gecko finger surface. All these special properties, these come from the nanostructure. So these are the two main ways that we can really use the nanotechnology in practical applications. Now, if you go into fertilizers, now when it's come to fertilizers, as we all know, the plants require certain nutrients for it to grow. We need carbon, oxygen, hydrogen, and these three are actually free. So we get hydrogen and oxygen from water, carbon dioxide from air. So these are free in many ways. But there are certain other nutrients that needs to be available for the plant growth, specifically nitrogen, phosphorus, and potassium. Typically, plants obtain them through soil. And there are also various other secondary nutrients and micronutrients that are essential for plant growth. Now, so if you select a specific nutrient, a plant would have an uptake, a demand curve. So demand curve is simply the requirement of a specific nutrient with respect to the growth period of a plant. Now, when the plant is small, it only requires certain amount of small amount of nutrients. Then it keeps going at a certain range. You need a consistent amount of nutrients. So this is called plant demand curve. Now, ideally, if you want to fertilize a plant, what you need to do is closely match this demand curve. So this green color, if you take this nutrient uptake curve as the green color, this yellow color line would represent, in ideal situation, if we can mesh it, that would be the ideal situation of fertilization. But as you know, most of the fertilizers that we use are in fact inorganic fertilizers. They have very high solubility, but when you apply them, we get a very high increase of nutrient concentration, especially when you apply them in splits. Very high concentration of nutrients, that's usually higher than the demand curve. And what will happen is the plant would not really absorb these fertilizers. and they count as excess, and basically this will lead to if inefficiencies in terms of fertilizer use. So ideally, on the other hand, if you apply less amount of fertilizer, then again the plant growth will be inhibited, or there may be deficiencies that may coming up. So you don't want that as well. So the ideal situation is actually to mesh exactly the demand curve, but it's not possible with the common inorganic fertilizers, with the common fertilizers that we use. And if you really dissect the curve of the demand curve, you can see there's a lag, initial lag, and then there's sort of a linear type of release. Then we come to a decay in release, and also then finally we have a zero order type of a constant type of release. Now, so what we can do is, using nanotechnology, we can actually utilize various nanomaterials to give these type of release, especially the lag release, the linear release, a decay, and a zero order release. So if you can make a fertilizer that can actually represent, actually produce this type of release, this where the holy grail of fertilizers really is. The typical release, however, is like this. We have very high concentration coming up with time, and this will go down very rapidly. So that's why we usually use multiple times of fertilization in a given crop cycle. Now, when it's come to, so basically the way of addressing this problem is through control release fertilizers. When it's come to control release fertilizers, there are various type of control release fertilizers. We have delayed release fertilizer, which has certain delay, and even after applying this fertilizer, there will be a delay in fertilizer release. We also have sustained release fertilizers. So sustained release fertilizers will have a initial release, and it will be sustained throughout a period of time. And also this the other type called zero order release, which basically produce a plateau in terms of the concentration. A very consistent delivery is obtained. And we have triggered release, zero order release, as well as step release. So these are different types of control release aspects that we can obtain. And let's see how we obtain this. Now, common strategy to make control release fertilizer, especially through nano materials, is actually having specific type of a barrier coating, or a barrier, a nano technology based barrier that that can actually produce specific type of release. So now what will usually happen is the nanotechnologists would venture into this area. They will come up with a specific type of a coating that will activate through a trigger. This trigger can be when you incorporate the soil to water, these fertilizers to soil. The trigger can be simply the addition of water, or can be microbial action, can be the pH change, things like that. So when the trigger happens, this coating will produce it basically react or co-work with water in a specific way, dissolved in the nutrients, and finally releasing them. So this the way how the coating reacts with or interact with water is how the nanotechnology is basically is become very important, and that's where the main the design aspect of nanotechnology comes in. So let's look at various type of release. Now we spoke about delayed release. In delayed release, we have an initial delay in terms of release. So there are two main ways of getting that. So most of the time nanotechnologists would work with a special nanomaterial. When you put it into the soil, this nano coating or nano layer will decay. So they will decay, but until it comes to a critical coating thickness, the inner fertilizer or whatever the nutrients that you have inside wouldn't release. But when it's come to a critical release, they start to release. It's one way of getting the delayed release. The second methodology is quite similar. So the nanotechnologies would use a nanopolymer or nano coating that it might work with. Interact with water or microbes in the in the soil and making small perforations, small cracks or defects in the layer. And these defects will be control defects so that when it comes to a critical value, the fertilizer will start releasing. So there are two methods of very common methods of obtaining delayed release. And also when it comes to sustained release, basically how we, in terms of getting, this type of a release pattern. So there are various techniques that we can use. There are two methods primarily. The first method is actually, having special coating, that's basically a semi-permeable type of coating system. Now, if you have nutrients inside, we have the semi-permeable membrane outside as a coating. When you put this fertilizer into soil, what will happen is the water penetration would occur. Now, when the water penetration occur, as we know, these fertilizers are inorganic, substance. They will dissolve inside this particular capsule, fertilizer capsule. And when these ions dissolve in water, there will be high osmotic pressure, and depending on the osmotic pressure, it will release. And this release curve can be quite, consistent, giving us a sustained type of release. The second methodology actually is based on a composite type of architecture, is designing composite type of architecture where you have different particle size distribution of coated nutrients entrapped in a solid matrix. So this solid matrix can actually, it has a controlled permeation towards water. So when you put it into these granules or this substance, when you put it in oil or soil, control penetration of water occurs, providing sustained release of fertilizers. These are two methods of getting sustained release. And also, interestingly, the first order release is basically a type of release that you would really like to see most, because if you take the demand curve, the highest, the larger portion of this release curve is actually zero or the first order type. Now, first order type diffusion is quite commonly observed or obtained using, playing with the coating thicknesses and their concentrations. As we know, the diffusion is primarily dependent on the concentration gradient. And the temperature, what you can do is you can change the thicknesses of various, these advanced nano coatings and their concentration, encapsulation concentrations, finally giving, ultimate cumulative release profile that is in the first order. So these are some of the, concepts or, different strategies and methodologies which very widely used in nanotechnology based fertilizer applications. And let me go to some SLINTEC examples now. At Sri Lanka Institute of Nanotechnology, we have been working with a specific type of slow release fertilizer, control release fertilizers. We have pioneered a method of making a biodegradable control release fertilizer to be used in primarily for paddy cultivation and various other, vegetables and all those of crops. So these are actually in testing the field levels testing scale these days. And also we have worked on specific method of making rock phosphate soluble. And rock phosphate is actually a type of mineral that has high content of phosphorus in it, but it's not readily soluble. So we have used a special thermal strategy in making this rock phosphate into a nano-sized particle, nanocrystals, and through that increase in the phosphorus solubility. And also we can use some of these concepts in various other sections, not only fertilization, but making a fungicide, pesticide, and things like that. And by increasing the surface area, because these particles are very small, we get a high activity, especially as you can see in this case, we have made nanosulfur based fungicide that works at a very low concentration compared with bulk counterparts. Like 300 ppm, we could see a wide variety of fungal inhibitions were obtained. So if we So these are some examples. And also when it's come to soil nutrient management, soil nutrient management is simply the way of uh, using fertilizers and various other soil amendments in in agricultural landscapes for crop fertilization. And typically, when it's come to soil nutrient management, there are four main concepts. We have to put right amount from the right source and in the right place at the right timing. The whole idea of soil nutrient testing to have this type of management, the most requirement is to have a very good way of soil nutrient analysis. Because if you go for a blanket recommendation that doesn't really take a look at the actual soil nutrient levels in soil, so this will usually lead to low fertilizer use efficiency. Now, when it's come to, so what is the challenge right now? There are soil testing, methods that are being developed. There are a lot of conventional testing methods that are usually laboratories practices. This would, take some time and cost you more money. To address this challenge, there are new ways. People are coming up with the portable chemical test kits that can be used in the field, and there are also rapid test kits for gardeners. But these systems still have their problems in terms of the accuracy, and they still cost high amount. And also these can lead to errors because usually these products they still require chemical handling by the user. So the amount of the regions that you measure, the amount of regions that you put in, and all these things really affect your measurement, which is still require some technical expertise in the field. Usual farmers might not have this expertise that can actually. This has basically, really reduced the use of these, test methods in the field. So, what we can do is now using nanotechnology, if we can come up with a very rapid method of assessing the soil nutrient availability. For an example, using a simple colorimetric method, we can really come up with a site-specific fertilizer recommendation for that crop. And this, I think, is a very interesting application, and a lot of research has been moved towards this objective. To do this at Sri Lanka Institute of Nanotechnology, we are very we are working with specific type of technology called nano or micro fluidics. Nano microfluidic is simply like so we all have seen microelectronic chips. We have electrical conductors, conductive pathways going here and there, connecting with various electronic components. Similarly, you can also design fluid pathways that are going through, very, different that can, cater different fluid interactions like mixing, reactions, heating, sensing, and all that. So what we can do is we can incorporate this sort of architecture into a small chip kind of a setup, giving you this very user-friendly, very small type of device that can actually give you a soil nutrient availability. And so so a lot of work has been done in this area, and SLINTEC is also doing some work related to this. And so these type of microfluidic devices can actually be used to, now for an example, now this work is actually represents similar device, a fluidic microfluidic device that give you the amount of ammonium, phosphate, silicate, nitrate, and nitric mix in soil extraction solution. So what they do is they exactly run the specific standard, but in a very miniature scale. So we can do that in using some of these microfluidic applications. And another interesting way of doing this is again using another method. Is a branch of microfluidics. It's called paper microfluidics. So paper, as we all know, it wicks and it can transport certain level of moisture. So this can be engineered in a very specific ways to make this standard reactions run on a paper plate, giving you colorimetric indications of different level of nutrients. So so these type of interesting work is being carried out specifically for soil related applications. Yeah, so with that I would like to conclude my presentation. We will take the questions later at the final session.
Yes, thank you very much, Ruchira. And you may remember I mentioned in my introduction earlier on that one of the activities we do is helping members understand the regulatory requirements connected to placing nanomaterials on the market. So I would now like to hand over to my colleague, Dr. Blanca Suarez Medina, who is our Director of Regulatory Affairs, and who will cover the specific aspect of regulation when it comes to nanotechnology in the agri-food sector. Blanca, the floor is yours.
Thank you very much, Chiara. Just a question. Do you see my presentation in presentation mode?
Not yet.
No? Okay, let me... now, okay.
Yes.
Okay. Thank you very much. I'll give you an overlook of the regulation of nanotechnologies in the agri-food feed sector. And now, due to time constraints, I will only focus on the European regulation, and I will just highlight those issues which are particular to nano, because otherwise you have to follow the standard regulatory procedures. So first of all, and thanks to all these other presentations that we had before, we can see how the rapid development of nanotechnology has opened the door for innovation in many industrial sectors, including agricultural production. And we have already seen how nanotechnology can contribute to improve pest management and crop protection, mainly, as we have seen in this last presentation, through the better efficacy of pesticides. And how is this achieved? We have seen before now, this enhanced efficacy can be achieved in different ways. We have seen now here how if we decrease the size of poorly soluble active ingredients, we increase their solubility and bioavailability. And another way is also to do encapsulation of active substances. And then we can control or slow target release of the ingredients. So these are all possibilities we have with nanomaterials and nanotechnologies and agriculture. Also, the previous speaker was talking about tracing and identity preservation, which is quite interesting, also an interesting application of nano in the pesticides. So I move ahead, and before continuing with my presentation, I would like to highlight 3 terms which I will mention along the presentation. So generally, each formulation of a pesticide will have an active substance which will be responsible for the properties of the plant protection product. But we may also have other substances called safeners, which are added to reduce or eliminate any phytotoxic effects. This is harmful effects towards the plant. And also we may have synergists as well, which are added to enhance the activity of the active ingredients. So it's good to have these 3 in mind, though we mainly focus on active substances. So basically in Europe, pesticides are regulated by the Plant Protection Products Regulation, which is 1107/2009. And unfortunately, this regulation does not provide a definition for nanomaterials. So it's difficult to regulate something that we don't have a definition for. Also, there is no label required to specify that we have a nano in our product, unlike other regulations like the biocides. And it mainly, for risk assessment, follows the EFSA guidance, which is mainly focusing on oral intake, being EFSA. All in all, of course, the regulation tries to prevent unacceptable effects on the environment or the human health, of human and animal health. Here, I also mentioned the biocides regulation because at some point we will get into it as well. And also REACH, because this is the overarching regulation in Europe for nanomaterials. So basically, the Plant Protection Product Regulation requires a pre-market authorization procedure at both two different levels. We have the EU level, which is mainly taken care of by EFSA as well. And this regards the active substance. But we also have a member state level for the whole product. So each member state must approve the whole product. Each member state concerned, okay. And it's also a prerequisite, of course, that none of the elements or formulation must not be included in the negative list, which is found in Annex 3 of the Plant Protection Regulation. And regulation, well, we should also take into account that if we are talking about nano, we may be, the regulation may be asking for a completely new dossier. Unless we can prove that the biokinetics of our products are the same as any other standard product. Otherwise, it accounts as a new product. So basically, the regulation includes a very thorough risk assessment procedure. And for this, we need to look into the life cycle of our pesticides. As you can imagine, the life cycle of a pesticide is very complex. From the time it is thrown on the fields, it undergoes a lot of, probably several transformations. It may biodegrade due to the action of fungi, bacteria, or plants. It may be hydrolyzed due to differences in pH, or photolysed if there is due to the sunlight effects. We may be facing sorption issues, and where fertilizer may be attached to the soil or to clay. Again, it may age as well. And of course, it will be translocated along different environmental compartments, such as water, maybe ended up in the aquatic life, or soil and back into the products, which account for food and feed products. So it is quite a complex issue. Now, since we have to do a risk assessment, I always like to highlight that the risk assessment is composed of two different elements, which are hazard and exposure, because sometimes we only focus on one of those, and that doesn't mean risk. For example, here I highlight the publication produced by Greenpeace in 2015, where they identified that supermarket apples have pesticide residues. And this is all to do with exposure. However, in this article, they don't mention threshold limits or concentrations. So even then, I mean, we don't have the whole picture at all. Another issue is risk. Here I highlight this report by the International Association of Research on Cancer, IARC, where they state that processed meat is a carcinogenic belonging to group 1. And this is a hazard endpoint. It's not a risk endpoint again. So for risk assessment, as I mentioned, we have hazard and exposure. When we look into hazard, we look at the characterization of the intrinsic properties of this particular substance. And this will lead us to the identification of possible hazard effects on human and the environment. So when we do this hazard assessment, we identify the important parameters such as ADI, which is the acceptable daily intake, ARFT, which is the acute reference dose. This means the amounts that can be ingested in a period of 24 hours, or the IOL, which is the acceptable operator exposure level. So these are different points that we get from hazard and exposure. Also, we need to work out the amount that is concentrated in the soil or in the crops, and also in food. And we have to differentiate between acute exposure. This means that we may have it just once, or chronic when we have it for a long time. So this is all can be found in the EFSA guidance. This is a general risk assessment for food and feed and pesticides as well. So I would like now to focus on nano. Here, I mean, you may not see this whole scheme, but I will take you through it. So according to EFSA, if we have nano, first of all, we should do a physicochemical characterization just to determine that we do have a nano in the end, and our product may have degraded otherwise. So first of all, they have a scheme which is based on the outcome of the project Nano Define, where one can see if we actually have nanomaterials in our products. Because if we don't, then we can just do a standard risk assessment for standard products. If we do have a nano in our product, then the second step is a recommendation to do an in vitro digestion exercise in vitro. And here we can revert to section 621 in the EFSA guidance, because the conditions in the gastrointestinal tract are very harsh. So maybe our particle does not survive. So if we don't have a particle after that, then again, we can go back into the standard guidance. Otherwise, we go ahead and do some in vitro testing, and then we also do stability studies on lysosomal fluid, again, mimicking what's happening inside the cells. If we still have a nanomaterial, then we should get into more in-depth characterization and with in vivo experiments. And this will give us our hazard characterization of the product, which will be combined with the exposure, and then we can do our risk characterization. So from here, we'll go now into exposure and hazard, following the EFSA guidance and focusing on nanomaterials. So first, I said we have to characterize our nanomaterial. And this is a list of parameters which will affect toxicity. So morphology is very important. It's not the same to have rigid or flexible tubes if we are talking about nanotubes. Then we also should look into the transformation of our nanomaterial, that is aging. Also to see if our nanomaterial is persistent or is actually highly stable. Also, well, there are several issues here, by accumulation, quantum effects, etc. So all these parameters have been associated with toxicity. Also, we should characterize our material, as I said before, to make sure we do have a nanomaterial. As you can see here, a list of parameters. And actually, the EFSA guidance gives some ideas of which technologies to use if we are dealing with nano. Again, these are those issues I mentioned before, which will be linked to our toxicity outcomes. So the EFSA guidelines highlight the stability, which is very important. Also solubility, of course, because if our particle is soluble, then we won't have a particle anymore. Dispersibility is very important, and also reactivity, because if our particle is very reactive, it may interact with the cellular components of DNA, enzymes, proteins, etc. Yeah, so now, as I said, risk assessment is composed of hazard and exposure. So this is the exposure flow chart that EFSA recommends. It's important to follow this flow chart because we may waive some experiments, and then we may get into a more cost-efficient evaluation. So the first thing we should do, as I mentioned before, by the way, CB and EFSA, here we focus on oral exposure. So the first thing is to define the type of our nanomaterial application. Is it an ingredient, an additive, a pesticide, a food contact material? This is important to determine. Then we should collect all the information on the characteristics of the pristine nanomaterials I already mentioned to you before. And also we have to see the amount of nanomaterial that we are adding into the food or feed item or food contact material. And from here we have two options, and we go into direct exposure, which will be via food or feed, or maybe indirect exposure, and this refers more to food contact materials to see if the material migrates out of the product, and then we are exposed. So depending on the outcomes of those two, we may just be diverted into a standard, the EFSA guidance for risk assessment. Otherwise, then we move forwards and then start, well, what we mentioned before, to see if our particle degrades in the digestive tract condition and the under digestive tract conditions. Yeah, so if it doesn't, then we do have a nanomaterial, and then we have to go and do the full characterization and exposure estimations to get our final data to fit our risk characterization scheme. And this is now regarding hazard. Again, we have to base our studies on the degradation of the nanomaterial under the digestive gastrointestinal conditions. It's also important to identify any population which may be may be more exposed to our product, maybe operators, workers, etc. And then from here, if our particle doesn't degrade under the digestive tract conditions, then we have to start retrieving data. It could be in the literature, we get information on the fish chem and the toxicity, but also we can generate our own data in vitro, which will tell us more or less what are the possible hazards associated with our nanomaterial. If we see that there are hazards associated with the nanomaterial, then we have to go into in vivo studies. In principle, the guideline recommends the 90-day oral toxicity study in rodents, and also we should do also in vivo genotoxicity studies. If we do collect information from in vitro studies which tell us that our nanomaterial is safe, then we may waive these in vivo studies. And after this, then we again gather the data for a risk characterization. So as I said before, the EFSA guidance focus on oral exposure, but they do have a few pages at the end of the document dedicated to pesticides. EFSA recognized 3 types of pesticides. We have the nanoparticulates themselves, or with a surface modified or pristine. We may have also nano-sized droplets in an emulsion or in solid lipid particles, or we have nano-encapsulated in natural or synthetic polymer shells. So these are the 3 types of pesticides that EFSA recognizes. In any case, also EFSA highlights that we do need a definition of nanomaterial under this plant protection product regulation. And the regulations should also mention that the approval of the active substance does not include the nanomaterial form unless explicitly mentioned. So as I said before, if we have nano, we may be dealing with a completely new product. So they do recommend to do a particle size distribution in the case of nano-encapsulated active substances, but not in the case of nano-formulated emulsions. And always to monitor the different behavior of our formulations, because a change in behavior may mean a change in the toxicokinetics of the product, and then we should do a risk assessment again. And regarding hazard, here you can see all the hazard endpoints we may need to do for a standard product. And in the case of a nanopesticide, the same applies, it's not different. But if we can prove that the behavior of our product is the same, we may just be able to do a read across exercise from the data we already have. Otherwise, they also recommend to introduce the OECD 407 guideline, which is a repeated dose 28-day oral toxicity study in rodents. But as I said before, the endpoints will be the same. The only trick here is to adapt our technologies to the nano size, because most of these guidelines were not developed for nanomaterials, but for fully soluble products. Besides this, the EFSA guidance also recommends to perform a dermal absorption studies, again adapted to the nano size, which could be very tricky. Also to perform inhalation exposure, as I mentioned before, this repeated exposure, and also to check the absorption through the gastrointestinal tract. Now, as I said before, all these guidelines were not developed for nanomaterials, but for soluble products. And we are aware now, and NIA is part of several initiatives which are actually adapting those guidelines to nanomaterials. So it's important to watch the field because it's moving really, really fast. And finally, just to end with the recommendation from EFSA, since the plant protection products doesn't have a definition for nanomaterials, they do recommend to divert to the biocides regulation for now. This definition is based on the European recommendation. However, it has the 2 particulars are not mentioned. First of all, the criteria referring to the specific surface area, which is under the European recommendation, may be used to determine a nanomaterial. This is not mentioned in the biocides regulation. And the biocides also doesn't mention that if we are dealing with particles of a particular concern, we may lower the threshold value lower than 50%. So these are 2 items which are not mentioned in the biocide regulation and deviate from the European recommendation. And with this, I think I stop now, and I will be happy to take any questions. I hope it was not too boring. Thank you very much.
Thank you, Blanca. Thanks a lot. We are just at tiny little bit behind schedule, so I am just going to conclude this public part of the presentation of the webinar by just mentioning that the Euro Nano Forum takes place next week. NIA has a satellite event on the 4th of May with the Nano Safety Cluster, and then the main event takes place on the 5th and 6th of May. And we have a couple of sessions in the context of our EU-funded project. So the event is entirely free of charge, and I hope to see many of you there. With that, I would like to conclude the open part of this webinar. Invite all those who are not members of NIA to kindly leave the